Hello, thank you for joining. My name is Pavel Princev, and I'm the Director of Business Development at Bridge Biotherapeutics. As a public company, the usual disclaimer is applied, and my remarks today may contain forward-looking statements. In this presentation, I will briefly introduce to you our company, as well as give a short introduction to some of the assets in our pipeline. Bridge Biotherapeutics is a clinical stage biotech company founded in 2015 with its headquarters located in South Korea and with additional offices and labs spread out across the United States and China. The South Korean branch oversees the clinical stage research and development of our core pipeline, sets the overall company business strategy, and leads BD initiatives that are focused primarily on the Asia-Pacific region. The Boston Discovery Center was established in October of 2020 and is located in Cambridge, Massachusetts. The center oversees new business development across North America and Europe, while also carrying out R&D work for their own discovery programs. We have 6 people in our senior management team. James Lee, the founder and CEO of Bridge Biotherapeutics, comes from a background in structural biology from Seoul National University, who has over 25 years of industry experience in research planning, and business development at LG Chem. Prior to founding Bridge Biotherapeutics, James has also founded and co-founded 2 other biotech companies in South Korea. Along with James, our well-balanced management team is comprised of experts spanning a range of disciplines, including scientists, entrepreneurs, financiers, and medical doctors, to name a few. As of this year, we are comfortably situated in our growth phase. The Boston Discovery Center is now fully operational with their own internal discovery pipeline. Over the past year, we have increased our employee headcount by 60%, and by the end of 2025, we aim to surpass 100 employees in our workforce globally. Additionally, we strive to increase our clinical pipeline from 3 to 7 programs through our active global BD in-licensing efforts, and within the next 4 years, we hope to have our first FDA marketing-approved drug. Finally, for the evolution phase and beyond, we envision to transform Bridge Biotherapeutics into a global stage company with an established sales network in the US with over 500 employees worldwide. Since January 2021, we have substantially expanded our workforce across the board. In order to keep up with the pace of our developing research projects, we have greatly increased the size of the R&D team. Similarly, we have also increased our BD workforce so that there is more personnel available to focus specifically on domestic and ex-Korea partnering, as well as our global search and evaluation activities. The strategy and management team has also grown by 50% over the last year. Our clinical stage assets include 3 first-in-class programs for treating ulcerative colitis, idiopathic pulmonary fibrosis, and non-small cell lung cancer, which I will go into more detail during this presentation. Additionally, our Boston-based Discovery Center has several discovery stage assets that are under development, both in-house and together with academic and industry partners such as the Scripps Research Institute and Atomwise. Starting off with BBT-401, we have developed a first-in-class polyno-1 inhibitor for the treatment of ulcerative colitis. BBT-401 inhibits polyno-1 via a novel mechanism of action and has shown to have a strong safety and anti-inflammatory efficacy profile in our preclinical studies, eventually paving the way for its entrance into the clinic. Given that the location of UC-affected tissue is deep in the GI tract, BBT-401 has been developed as an oral formulation with a gut-restricted distribution profile that minimizes systemic exposure and increases site-specific proximity of the drug. With this formulation, we have obtained an excellent phase 1 clinical safety profile and are now evaluating the efficacy of the drug in the phase 2A trial. For this trial, the low-dose cohort has been completed, and we are now currently enrolling patients in the mid to high-dose cohorts across several sites in Korea, the US, New Zealand, and in Eastern Europe. The left-hand side graphic illustrates the novel MOA of BBT-401. It is well known that toll-like receptor signaling plays a key role in the pathogenesis of several inflammatory diseases, with UC being one of them. BBT-401 is a protein-protein inhibitor that disrupts this aberrant signaling pathway by preventing the formation of the polyno-1 containing MyD88 and RIP1 complexes. By doing so, downstream NF-kappa beta is unable to translocate into the nucleus and initiate the induction of pro-inflammatory genes that lead to the formation and exacerbation of ulcerative colitis. In the middle preclinical efficacy chart, BBT-401 was administered to mice with DSS-induced colitis at 3 dosages. We were able to see a clear dose-dependent effect across the 3 dosage levels, and when compared to a competitor, the medium and high dosage groups showed a significant reduction in disease severity. On the right-hand side table is some key clinical efficacy data of our low-dose cohort from the phase 2A trial. Over an 8-week period, patients were given our gut-restrictive oral formulation of BBT-401, upon which drug efficacy was measured using the industry-standard Mayo score. Our drug has shown to reduce the Mayo score in several of the treated patient groups, as well as reduce the presence of a key disease biomarker when compared to control. With the low dose cohort portion of the phase 2A trial completed, we are now dosing patients in the medium to high dose cohorts. This is being conducted globally with trial sites in the US, Korea, New Zealand, and in Eastern Europe. Last October, we initiated the phase 1 China ethnobridging study in healthy volunteers, which has successfully completed in January without any noticeable concerns. A detailed clinical study report is expected to be available in the second quarter of this year. Our next clinical asset is BPT-877, a first-in-class autotaxin inhibitor for treating IPF and other fibrotic diseases. Idiopathic pulmonary fibrosis is the most common form of idiopathic interstitial lung disease. It is a devastating lung disease that progressively worsens over time. The disease induces scarring and inflammation in the lung, interfering with the patient's ability to absorb oxygen from the air. The current standard of care varies with the severity of disease. Treatment options typically include supplemental oxygen, symptom management, lifestyle changes, and in some cases, treatment with antifibrotics. The existing treatment options, however, are still largely inadequate, with about 70% of patients dying within the first 5 years of diagnosis. BPT-877 has successfully completed its phase 1 studies, and we are currently in discussions with the FDA for planning the initiation of the phase 2 trial. On to a few words on the MOA and some key efficacy data from the phase 1 trial. Autotaxin is an enzyme that has been found in higher concentrations in patients with inflammatory and fibrotic diseases, as well as in tissues of patients with fibrosis of the lung. Autotaxin catalyzes both the hydrolysis of LPC and the local production of LPA. In turn, that LPA then binds with and activates several LPA receptors, triggering a downstream signaling cascade that leads to epithelial apoptosis, the primary initiating pathogenic event in mottled pulmonary fibrosis and IPF. Furthermore, LPA has been associated with promoting inflammation and pulmonary edema, as well as other pathogenic events that further exacerbate the disease. The selective binding of BPT-877 to autotaxin effectively inhibits autotaxin's ability to catalyze LPC and promote LPA production, greatly slowing down the progression of the disease. The chart on the right displays the LPA levels in patients receiving BPT-877 in a multiple ascending dose from our phase 1 trial. It can be seen that for 800 milligrams QD dosing, a greater than 80% LDA inhibition was observed at steady state levels when compared to baseline. For BID dosing, 80 and 90% LDA inhibition levels were achieved at the 100 and 200 milligram dosages, respectively. With the phase 1 trial successfully completed, we are now in discussions with the FDA for our phase 2 trial design and plan. Additional animal studies that were requested during our last FDA Type C meeting have been completed, and a follow-up meeting with the FDA is scheduled for this summer. We anticipate initiating the global phase 2 study shortly afterwards. Our final clinical stage asset is BPT-176. It is a first-in-class, fourth-generation EGFR TKI for the treatment of non-small cell lung cancer. NSCLC is the largest subset of lung cancer that accounts for 85% of all lung cancer cases and is also the most common cancer with the highest death toll worldwide. Due to the aggressive nature of the disease, the average 5-year survival after diagnosis is often below 5%. One of the most well-known drivers of NSCLC are mutations in the EGFR gene. Previous generation EGFR TKIs target such mutations and have shown promising results, particularly the third generation EGFR TKI blockbuster drug osimertinib. Unfortunately, the majority of patients taking osimertinib stop responding to it because they have developed new mutations in EGFR, which prevents the drug from working properly. This key resistance mutation has been identified as C797S. Patients with C797S mutations are left with limited treatment options, making the development of a next-generation EGFR TKI that is active against the C797S mutation incredibly important. BPT-176 has shown outstanding preclinical safety and efficacy when compared to existing and developed products. Given its excellent preclinical profile, BPT-176 has become the world's first fourth-generation EGFR TKI to enter the clinic, with the first-in-human dosing being completed in April of last year. To ensure that BPT-176 is provided to patients who stand to benefit it from the most, we have an effective biomarker strategy in place, which enables us to screen, identify, and closely monitor patients' EGFR mutation profile by a liquid biopsy through the entire course of their treatment. This slide summarizes the biochemical and cellular activities of BPT-176. We conducted a head-to-head comparison of our compound against the third-generation osimertinib as well as the first-generation gefitinib. As can be seen in the top panel, summarizing the cell-free biochemical assay data, BPT-176 exhibits broad and highly potent single nanomolar activity against single driver mutations, as well as C797S containing double and triple EGFR mutations, all while sparing wild type. Moving on to the cell-based assay data, we see that BBT176 remains consistent with what was seen in the biochemical assay. It is active in single, double, and triple mutations with heightened potency towards del19 containing mutations. In both assays, BBT176 shows weak activity against wild-type EGFR. Here's a summary of our in vivo experiments. We tested BBT176 across a wide range of cell and patient-derived xenograft models, as well as a brain metastasis model, all of which showed clear tumor regression or inhibition. In this particular experiment, we were interested to see how BBT176 responds in a PDX model of a patient who has been through 7 lines of therapy, including chemotherapy, erlotinib, osimertinib, and has been found to carry a DDC triple mutation. We were able to see a dose-dependent anti-tumor effect with tumor regression achieved at 100 mpk administered once daily, all while having a minimal effect on body weight change. Moving on to the trial design, we are now in the 5th cohort of the Part 1 dose escalation study, which is being conducted across 3 sites in South Korea. Patients enrolled in the Part 1 study have presented with locally advanced or metastatic non-small cell lung cancer with a confirmed EGFR mutation. Patients undergo liquid biopsy at screening, as well as at regular intervals throughout their treatment, so that their EGFR mutational profile can be closely monitored as treatment progresses. Currently enrolled patients have been confirmed to carry single driver, double, and Tagrisso-resistant triple mutations. With Part 1 expected to be concluding this year, we will be initiating the Part 2 dose expansion trial in Korea and the US, focusing on patients with confirmed C797S containing triple mutations. Looking at our expected timeline, interim results from the Part 1 study are planned to be announced at ASCO this year, where I hope to see you there. The Part 2 dose expansion study has been designed to allow a path for accelerated approval, for which we expect to initiate the trial towards the first quarter of next year, shortly after our end of phase 1 meeting. BBT207, which is currently under IND-enabling preclinical development, is an internally discovered 4th-generation EGFR TKI with demonstrated potent preclinical activity against a broad range of the most common EGFR mutations prevalent in non-small cell lung cancer. BBT207's preclinical activity profile allows it to be positioned in earlier line settings for 1st, 2nd, and 3rd-generation EGFR TKI-resistant patients. While BBT176 is exceptionally active against Tagrisso-resistant DTC and LTC triple mutations, BBT207 was developed with the goal of providing treatment options in earlier line settings, such as for patients who harbor either T790M or C797S containing double mutations that arise from the progression on earlier generation EGFR TKIs. Our preclinical data suggests that BBT207 may also have the potential to be positioned as a frontline treatment as well. For our development timeline, we expect to submit the IND towards the end of this year and initiate phase 1 clinical trials by early next year. Also, please keep an eye out for us at this year's AACR event in April, where we will be giving a poster presentation highlighting our preclinical data on BBT207. Our company has shown solid financials. These are some of the numbers at a glance. Our current market cap as of mid-February is $198 million with total assets valued at about $43 million. We also have a strong cash runway until the end of 2023. Our stock price has been somewhat less steady over the past year. However, we believe that our differentiated strategy exploiting global science to address clear medical needs in combination with the potential milestones coming from our 3 early to mid-stage clinical studies will start to get reflected in the broader market. To summarize, we have 1 multinational phase 2 study ongoing for ulcerative colitis therapy, and an additional 2 phase 2 studies planned to begin this year for non-small cell lung cancer and idiopathic pulmonary fibrosis. All 3 of our clinical stage assets are first in class and are leading the way to bringing much needed medicine into the clinic, which will address substantial unmet needs in their respective indications. And finally, we have multiple in-house discovery and preclinical stage assets that are currently under active development. With the rapid growth of our company during the last year, we continue to actively expand our internal R&D pipeline, both in Korea and the United States, through new discovery efforts and open innovation, particularly from within the Boston Discovery Center. In the long term, Bridge Biotherapeutics is on track to becoming a fully integrated bicontinental biotech company. This concludes my presentation on Bridge Biotherapeutics. If you have any questions or would like to know more about our programs, please send us an email. We look forward to hearing from you. Thank you.